So this paper is top-down perspectives on cell membrane potential and protein transcription. And this is a paper by Javier Cervera, Michael Levin, and Salvador Maffei. This is in Scientific Reports. This is a new paper on thinking about these levels of analysis and thinking about different problems in biology. So this one is on cell membrane potential and protein transcription, but approaching it from more of a top-down perspective or at that higher level of analysis. So we could spend a lot of time, and people do, thinking about the molecular biology of this. And the question is that strictly molecular biological approach useful to the problem, or can we get a new perspective by going up a few levels of analysis and looking at the problem? We're looking at how these things are controlled maybe by top-down mechanisms. So the abstract reads, we have explored a simple model for the multicellular interplay between bioelectricity and protein transcription using a top-down perspective that offers new insights complementary to the commonly used bottom-up descriptions. So again, a lot of ways in which we understand cell membrane potential and protein transcription are through these bottom-up descriptions. It's like, okay, you have a synthesis of different molecules, and those molecules interact, and we're very detailed in our descriptions of that, and it is obvious that that is the mechanism. But that's, of course, not obvious, nor is it even really maybe the right answer. We model the non-excitable cell bioelectrical representation of the external environment, including the neighboring cells, using voltage-gated ion channels and intracellular junctions. So they have a cellular model, but it's very different from what they were doing with CompuCell 3D in their cellular POTS model. They're actually modeling cells as, or populations of cells, as these ion channels and intracellular junctions, and then building a network out of that. The simulations make 3 predictions. Okay, so they're simulating this with their cell representations or cellular representations, and from these simulations they make 3 predictions. The first is that shifts in membrane potential, which you can model if you model this dynamically instead of statically. Shifts in membrane potential allow transitions between gene expression states. So we get these sort of native Markov states, basically, that we can extract from the biological data. Number 2 is in the case of different cell potential gated transcriptions, depolarized cells cannot control the distinct gene expressions as effectively as polarized cells. So we can see a difference in how the state of the cell itself controls how this process works. And we can only do that through a model of the cell and of neighboring cells, because that controls the state of the cell. And 3, community effects should permit to extend the single cell control to the multicellular level. So again, community effects are the cells surrounding the focal cell that you're interested in. And thinking about that population of cells as a network where cells are affecting their neighbor cells and so forth. This allows us to have this single cell control mechanism that transcends single cell constituents. Put it another way, if you use a model of molecular biology and look at sort of these molecular explanations as the sort of ultimate explanation, you'll miss a lot of products of cells interacting, of things happening at the cellular level. You'll completely miss those, and they won't be included in your model, and then your model can't predict certain things. Because the spatiotemporal distributions of instructive signaling ions and molecules depend on the local electric potentials, different multicellular potentials correlate with distinct downstream gene expression patterns. So again, these multicellular potentials, which you get at the cellular level, actually correlate with gene expression patterns at the genomic level, at the molecular level. So we know that this is actually quite useful and actually explains a lot of things happening at the molecular level, and we don't have to worry about detailed modeling of that level because our model at the cellular level is capturing a central cell, which is the cell maybe of interest that we have, and then there are cells that are neighbors that interact with it, which I called the focal cell, but they call the central cell, is able to measure the number of neighboring cells and learn their bioelectrical state from the downward induced membrane potential changes, which suggests that multi-scale bioelectricity can operate as a top-down mechanism in development and regeneration. This is kind of like the same approach. Well, it's actually kind of an approach you might use or might be analogous to the whole model of morphogenesis or chemical morphogenesis. So with positional information, remember I said that was a way to describe morphogenesis, to describe pattern formations in cells. And one of the things cells need to know is their position. And the traditional Wolpert argument is that the genome of a cell is the source of that information, that it basically knows which Hox cluster it's a part of. It expresses genes in a certain way to create a gradient or to be a part of a gradient. And then that information is measured by sort of a difference from other cells neighboring to it. So that's how that explanation goes. In this case, what they're saying is that the cell itself can measure the neighboring cells, learn bioelectric states, and they do this not from a molecular mechanism, but from comparing membrane potentials. And so that gives you a mechanism that's very similar to that at a higher level of analysis. Now the question is, maybe there are other mechanisms that we're not, looking at here, and that's fair. But again, we have this model that kind of makes some assumptions about the molecular milieu, and there are a lot of papers on that, so we kind of know those mechanisms that are worked out. We can then focus on the next or higher levels of complexity, higher levels of organization, and then we can merely get correlations with those molecular states, and we can have a predictive model from that. So they talk in this paper about how in biological phenomena, causation is commonly analyzed from the parts to the whole. So that means that in our bioelectric world here, molecular biology is sort of, is composed to produce these cellular states. So it depends on what the molecular state is, and we just sum those together, and we can determine the cellular state. But of course, the problem with that is there are things happening at the cellular state that are independent of that, as well as kind of constraining what happens in the molecular world or the molecular states. So there's this interaction that we can't account for if we just focus on one level of analysis. So they start talking about top-down or downward causation, which is where you have this thing happening at the, say, cellular level that's affecting the molecular level. And if you have interactions between cells, the states of other cells can indirectly affect what's happening at the molecular level of a focal cell, say. Okay, so they kind of get into this. Here we have considered a simple model for the interplay between bioelectricity and transcription in non-excitable cells that explores a top-down perspective. To this end, we invoke an experimentally based model of bioelectrical adaptive transcription that involves feedbacks in multiple size scales, emphasizing the central biological role of the cell. Okay. And so they're kind of getting at this idea of multicellular patterns and biofields, as they call it. I guess they mean like bioelectric fields, but they kind of shorthand biofields. And this sort of approach can benefit from descriptions at scales higher than the underlying molecular events, because top-down control offers new opportunities for external actions. In particularly, multi-scale bioelectricity constitutes a high-level spatial scale. So we have this aspect of the spatial scale between cells across an array of cells. And of course, that array of cells is heterogeneous. So some places in space are different from other places in space. Maybe that's because it's part of a different tissue. Maybe it's because there are constraints on different places in the tissue. And again, like as Susan points out, there's always a mechanics to worry about here. And so this will at least give us some insight into that variation. So multi-scale bioelectricity constitutes a high-level spatial scale control node that expands from the subcellular membrane potentials of organelles, such as lysosomes and mitochondria, to the spatial temporal patterns of multi-cell potentials characteristic of development and regeneration. So this is where we're going from things going on at the organelles, to the cells, to neighboring cells, to ultimately tissues and then organisms. And this allows us to do these kind of large-scale things like development and regeneration. So an organism relies on these things. Michael Levin has done a lot of work in flatworms and looking at regeneration in flatworms. And flatworm regeneration is interesting because it's totipotent, meaning you can take a single cell from a flatworm and you can plate it in a new environment, and it can grow an entirely new flatworm from that one cell. And that means that's because each of the flatworm cells is totipotent, meaning that one cell can produce an entire organism or reproduce an entire organism. And so the thinking of that whole line of work is that this is driven by bioelectricity, that as this one cell reproliferates, there's a bioelectric field that forms, and that bioelectric field is information about what the pattern should look like. So this is like this pre-patterning idea that they stuck into the Wolpert model, where they had to kind of come up with a stopgap explanation for why you have these larger scale patterns. In this case, it's more detailed. You have these bioelectric fields that get coordinated, and so now you have to find a mechanism for that, and that's what they're doing here. We consider here the coupling between bioelectricity and the transcription of two model proteins. To this end, we operationally describe the internal cell representation of the external environment, including the neighboring cells, using voltage-gated ion channels and intracellular junctions. This bioelectrically centered model is complementary to the traditional descriptions focused on biochemical networks and biomechanical fields. Okay, so this is when they get into their model. They get into the membrane potential as their crucial parameter that defines the electric potential difference between the cell inside and the external microenvironment. So you have these easy to define parameters that you can use in your model. They're already used in the experimental models, so they're just used in the computational physical model as well. And so they talk about sort of how membrane potential works to polarize and depolarize cells. While crucial information of the body plan resides within the DNA as a basic single-cell language for making proteins, multicellular potentials can transduce short-term bioelectrical information into long-term transcriptional processes because of the coupling of the signaling ions and molecular concentrations with the local electrical potentials. So in this way, the spatial temporal patterns of cell potentials can provide downstream biochemical information for the establishment of different morphological regions. So you see that there is this voltage or this membrane potential, V, that is really giving you an indicator of the cellular state. And this V is connected then to the molecular world in that it's descriptive. It's a shorthand for a lot of protein synthesis processes, other types of transcriptional processes. We don't need to be too precise in those because all we're really interested in is how V fluctuates and how they contribute to V, the value. And we can look across cells and see membrane potentials for those cells. And then we can ask questions like what happens if membrane potentials synchronize or desynchronize, or if there's long-term depolarization or long-term polarization, or things like that. And then we can go back and infer that there's certain molecular states that cause this in single cells. And so this is all kind of reminiscent, actually, of differentiation waves and some of the mechanisms there that have been proposed by Dick and his co-authors. So yeah, this is definitely that same type of model. And so this kind of goes through a little bit more about their model. This just shows an example of, where you have, say, a depolarized cell being part of a polarized neighborhood, and it has an effect on that neighborhood or on that cell. So this is the same type of thing we think about in cellular automata, where if you have, say, a cell of a certain state and you embed it in other neighboring cells, that it affects its state. So in this case, in A, a depolarized cell is embedded in a polarized neighborhood. So all these cells are polarized with respect to their V value. And in this case, this cell is depolarized with respect to its V value. And then if you put it into this neighborhood, it shifts the behavior of the focal cell to a polarized state. The same thing happens in a depolarized neighborhood. If you put a polarized cell into a depolarized neighborhood, that polarized cell will become depolarized just by virtue of being surrounded by those types of cells. And this is very similar to the types of things you see when you take, say, like a stem cell or a precursor cell, and you embed it into a bunch of differentiated cells. Like if you take a cardiac stem cell and you embed it in cardiac muscle, that cell, theoretically at least, becomes cardiac muscle because it's surrounded by those signals, and then it takes on that state, or it differentiates into that state. So then they just propose a model here where they have genes, proteins, those things through transcription and translation contribute to voltage-gated channels, which are part of the cells that define cellular state. There's a cellular potential regulation or transcription, so the potential itself, if it's changing in the way that we saw in A and B, it can control sort of the rate of transcription for certain proteins. And then the cellular aggregate has this community effect in controlling the states of single cells, and cells themselves communicate through gap junctions or electrical connections between them that regulate the polarization state of the cell. So these cells not only exist as neighbors, but they actually are communicating with each other and sharing information about their state.